Hello my dear students, this is Vasavi Ma'am from Neet World, your biology educator for Foundation and CBSE. I welcome you all to the most interactive and interesting live session, children. Yes, in the previous class we have just started the discussion and we have just started the topic of endomembrane system, and we have completed the structure and functions of the nucleus, and then next we have also learned in detail about the endoplasmic reticulum, children. Okay. So, once if we recall about the previous session what we have discussed about the endoplasmic reticulum. So, the endoplasmic reticulum is going to be in two forms, that may be either smooth endoplasmic reticulum or rough endoplasmic reticulum. The smooth endoplasmic reticulum is involved in the formation of lipids, it is involved in the synthesis of lipids, and then next the rough endoplasmic reticulum is involved in the synthesis of proteins. This is what we have discussed in the previous session. So, coming towards the today's particular session. So, children, students, can you remember I have gave you one question to all of you, that which cell organelle is called as the postman of the cell. Here comes an answer for that, children. Yes. So, that is Golgi apparatus. The Golgi apparatus is called as postman of the cell. The Golgi body is called as the postman of the cell. So, what is the reason why we are calling that Golgi body as a postman of the cell, children? So, if you observe in our daily life, when we are going to place an order something, first the delivery boy, okay, so first the person who is going to take the order, first he is going to receive the order, and then next he is going to modify, then next he is going to pack that particular order, and then next he is going to distribute that order to the particular destination. This is what we are going to see in our daily life. This is just an example, children. Okay. So, whenever we are going to order something, or whenever we are going to give gifts to someone or someone else, so then we are going to just take the gifts, or we are going to receive the parcels, and then next the person is going to pack the particular order, and then next he is going to distribute that particular product in a particular destination. So, it is nothing but receiving the products, then next modifying, that is nothing but packing, and then next distribution, that is nothing but then next dividing it, or else then next sending it to a proper destination. This is the actual process, right? Yes. So, the same function is going to be performed by the cell organelle called as the Golgi body. Okay, the Golgi body is also involved in receiving the substances, receiving the substances like proteins, and then next it is going to be undergone the process of the modification, and then next it is involved in the secretion, packaging, modification and secretion. These three are the most important functions which we can observe in the Golgi body. The Golgi body is a cell organelle, that is why it is called as the postman of the cell. It receives the proteins, it modifies and it packs the proteins, and then next it is going to secrete the proteins. So, let me explain you in clear way. Yes, children. So, if you look into this small animated video, you will get the idea. This is the how actually the Golgi body is going to be present in our body. See, can you see here? Yes, from here the proteins are going to be, it is going to be received, and then next here the modification is going to take place, and then next it is going to receive. So, now we will learn it in a detailed, children. Yes. So, Golgi body was discovered by the scientist Camillo Golgi in the year 1898. The Golgi body was discovered by the scientist Camillo Golgi in the year 1898, and the structure of the Golgi body, the cell organelle of the Golgi body is going to contain three most important structures, or else three most important components. Those are cisternae, vacuoles and vesicles. If you look into this picture, you will get an idea, children. So, the cisternae are the flattened disc-like structures. The cisternae are very flat in nature and they are disc-like structures. Can you see this? These are the cisternae which are stacked and which are connected to one another, and these are very flat in nature, and these are the disc shaped structures which are going to be remain in close association with one another. So, in this way many cisternae are arranged in order wise. Okay, and this is about the cisternae. And then next vacuoles. The vacuoles are the spherical shaped bodies. So, if you observe the difference, the small minute difference between the vacuoles and vesicles is, see, the vacuoles are located by joining to the cisternae. The vacuoles will join the cisternae and they are going to be bulged outside. See, if you see here, here we can find one vacuole. Can you see this? And the vacuoles are located towards the edges by joining to the cisternae. The vacuoles are involved in storing the substances, children. So, here what we are going to transport? We are receiving the Golgi body is actually receiving the proteins and it is going to modify it, and the next it is going to secrete it. So, in between this process, the vacuoles are going to arise which will attach to the cisternae, and these are also the spherical shaped structures which will store the substances like proteins or else sometimes lipids. Yes. So, here are the vesicles. So, in between these Golgi apparatus, randomly here we can see the vesicles. So, the vesicles are small membrane bound structures. These are the small membrane bound sacs which are involved in storing and which are involved in just transporting the substances, children. The vesicles are involved in transporting, and the next vacuoles are involved in storing the substances like proteins. So, now if you see, this is how actually the Golgi body is going to look like, and these are the structures which we can observe in different forms like cisternae, vacuoles and vesicles. So, the cisternae are flattened disc shaped like structures, and the next vacuoles are the bulged spherical bodies which are going to be attached towards this cisternae, which are involved in storing the proteins, and the next vesicles are small membrane bound sacs which are involved in just transporting the substances like proteins. So, now if you look into this, here we have two phases. We have two different functional and structural compositions like cis phase. Yes. So, this is called as cis phase. Cis phase, or else it is also called as the forming phase. And the next here it is called as the trans phase. Trans phase is also called as maturing phase. Cis phase and trans phase. So, now when we have studied the endoplasmic reticulum, the endoplasmic reticulum is also going to be present in two forms, that may be either rough endoplasmic reticulum or smooth endoplasmic reticulum. So, if I ask you a question where the proteins are going to be synthesized, absolutely the answer is going to be the rough endoplasmic reticulum, students. So, in the rough endoplasmic reticulum, the proteins are going to be synthesized, and that proteins are going to be travelled, okay, that proteins are going to be transported to this Golgi body. That proteins are going to be stored in the vesicles by the help of these small vesicles. The proteins which are synthesized in the rough endoplasmic reticulum, they are going to be transported towards these vesicles, and these vesicles will carry that proteins to the cisternae present in the Golgi body. Clear with the concept? So, actually I have gave you one term, that endomembrane system. So, actually the endomembrane system includes the nuclear envelope, and then next the small vesicles, and next Golgi body, endoplasmic reticulum, and the next lysosomes. This is what actually we are calling it as endomembrane system, students, because all the functions are going to remain in close associated. All the functions are going to communicate with each other to provide the better functions for the cell. So, that is what we are calling it as endomembrane system. In the endomembrane system, different cell organelles like nuclear envelope, endoplasmic reticulum, next vesicles, Golgi body and lysosomes, these all are going to be coordinated and communicated with each other to provide the to perform the functions. So, yes. So, these are actually the proteins which we are getting it from the rough rough endoplasmic reticulum, rough endoplasmic reticulum. Yes, rough endoplasmic reticulum. Yes, from here we are going to get the proteins. Okay. So, due to the presence of ribosomes, the endoplasmic reticulum is called as the rough endoplasmic reticulum, and here the proteins are going to be synthesized, and from here the proteins will travel, and the proteins are going to be taken by the small vesicles. The vesicles will receive the proteins, and then next the proteins are going to be distributed to these particular cisternae. So, now if you see this cisternae, it is called as the cis phase, or else it is also called as the forming phase. Why means the cis phase is going to be remain in close association with the endoplasmic reticulum. The cis phase is going to show the face towards the endoplasmic reticulum. It will just receive the proteins, and then next inside each vesicle the protein is going to be embedded, and in these cisternae, in these all the cisternae, the protein is going to get modified. First it is the Golgi body is going to receive the proteins. After receiving the proteins, in each cisternae the modification of protein is going to takes place. I will make you this concept crystal clear by showing you one animated video, children. So, until then just please try to understand the concept whatever I am teaching you. Yes. So, these are all the cisternae, and in each cisternae the protein is going to get modified. It will modify in the first cisternae. After modification in the first cisternae, next it is going to be delivered to the second cisternae. From the second cisternae, it is going to be delivered to the third cisternae, fourth cisternae, and so on. So, later it is going to be released. The modified proteins are going to be released from the trans phase. It is also called as the maturing phase. So, these are the secretory vesicles, and then next the vacuoles are involved in storing, and these vesicles are involved in secretion. The first vesicles whichever they are going to be stored, first it will receive and then next it will secrete. So, this is what actually the main components which are involved in the Golgi body. Hope this concept is clear to all of you. So, the Golgi apparatus is called as the postman of the cell. This is just an example to all of you. So, whenever we are going to order something, first the person is going to receive the product, he is going to pack it, and then next he is going to send it to the proper destination. This is what actually it is going to happen. But when it comes to the Golgi apparatus, here actually the proteins are going to be modified and secreted. So, in this process there are some small vesicles which will receive the proteins. After receiving each protein is going to get modified in each cisternae. So, then later on that proteins, the modified proteins are going to be sent out with the help of the secretory vesicle. Yes, now look into these points. See, the Golgi body helps in the secretion of mucus, enzymes and hormones. Yes, proteins are made up of the amino acids. So, the amino acids are also involved in the process of making the enzymes. It is involved in process of performing and process of doing the enzymatic activity. And next, modification, packaging and secretion of vesicles. This is the most important function which is done by the Golgi bodies. And next, it also helps in the formation of lysosomes. You will learn about the lysosomes in the upcoming slide itself. I will explain you there in a clear way, children. Yes, and next, it is also involved in synthesis of cell wall and plasma membrane. Okay, these all these cell organelles whatever we are going to study now, these endoplasmic reticulum, Golgi body, lysosomes, these all are actually present inside the cell itself. So, these all organelles are present in the cell, and sometimes it is also involved in the process of making the plasma membrane also. This is about the just a clear concept of the lysosomes and Golgi body. Yes, now we will discuss about the ribosomes. So, before discussing about the ribosomes, once again I will show you the short video clip of this Golgi body, how actually the proteins are going to be modified. So, students please look here. See, yes, I will play it. See, if you see here the pink colored structure, these are what? These are the proteins which are going to be released from the rough endoplasmic reticulum. So, after receiving from the proteins are going to be delivered from the rough endoplasmic reticulum. Yes. So, it is going to be sent to the vesicle. See, can you see here? Yes. Yes, it fuses with the vesicle, and the next in each cisternae it is going to travel and it is going to modify. See, first it will receive, the secretory vesicle will receive the proteins, it will secrete out, and again from the outer side it is going to enter into this particular cisternae where the modification is going to take place. After modification it is going to be sent out. Clear with the concept? Once again I will play, just watch this clearly. See, the proteins we are getting it from the rough endoplasmic reticulum. Yes, vesicle is formed, it is taking, and the next it is sending it to the cisternae. Yes, again in the each cisternae, in the next cisternae again modification is going to take place. Yes, here after modification this particular modified protein will release from the maturing phase, that is nothing but the trans phase. This is how actually the process is going to takes place. Yes, now coming towards the ribosomes. Yes. So, the ribosomes are the non-membrane bound cell organelles. These are the non-membrane bound cell organelles, and the ribosomes are discovered by Robinson and Brown in the year of 1953 in plants. Robinson and Brown discovered the ribosomes in plants. Later, another scientist, Plastids, in the year 1955 discovered the ribosomes, observed the ribosomes in the animal cells. So, these are actually, look into this statement, these are actually the membraneless ribonucleoprotein particles. These are the non-membrane bound structures which are made up of a protein called as ribonucleoprotein. Yes, and if you see the structure, if you see the ribosomes, we have two types of cells, prokaryotic cell and the eukaryotic cell. Yes children? Yes, we have prokaryotic cell and eukaryotic cell. So, in the prokaryotic cells, already you have discussed and you have learned in detail about the prokaryotic cell structure. Okay, if you once recall the diagram, there you can understand clearly. In the prokaryotic cells, the ribosomes are freely suspended inside the cytoplasm. The ribosomes move freely, and the ribosomes are embedded as a granular structures in the prokaryotic cells. They float freely in the cytoplasm. But when it comes to the eukaryotic cells, inside the eukaryotic cells, the ribosomes remains attached as a granular structures to the rough endoplasmic reticulum. This is going to give you a clear idea how actually the ribosomes are going to be present inside the cells children. When it comes to prokaryotic cells, they just move freely in the cytoplasm as a granular structures, but when it comes to the eukaryotic cells, they remains attached to the endoplasmic reticulum, that is called as rough endoplasmic reticulum. Due to this presence of ribosomes only, we are categorized that endoplasmic reticulum as rough and smooth. Yes. So, but here we have two types of ribosomes, one is prokaryotic ribosome and the next eukaryotic ribosome. So, in the eukaryotic ribosome, we have two units called as large subunit and small subunit. Clear? The larger subunit in the eukaryotic cells is made up of 60 S. Here the S is going to indicate, the S is indicated as a sedimentation coefficient. I am writing it here children. Yes, sedimentation coefficient. Yes. So, the S is the sedimentation coefficient in the ribosomes. In the eukaryotic cells, the larger subunit of the ribosome is made up of 60 S and the smaller subunit is made up of 40 S. Clear? So, together we are giving the sedimentation coefficient for this eukaryotic cells is 80 S. The 80 S type of ribosomes we can find only in the eukaryotic cells, but again it is made up of two units called as larger subunit and smaller subunit. The larger subunit is made up of 60 S type of ribosomes and the smaller subunit is made up of 40 S. So, together for these eukaryotic cells there is a standard sedimentation coefficient that is called as the 80 S. So, students, you might have get the doubt that this 80 S is not going to get by adding the larger and smaller subunits children. Okay, 60 plus 40, 60, 70, 80 and 90, 100. Okay, this is not the actual process of the sedimentation coefficient. This 80 S is going to indicate that during the cell extraction process, during the centrifugation process, we are going to get the standard residual structure, that is only called as the 80 S type. Okay, there in the centrifugation process it is identified as only 80S, only 80S type of ribosomes are going to be present. When it comes to the prokaryotic cells, 70S type of ribosomes we can find. In these prokaryotic cells, the larger subunit is made up of 50S and the smaller subunit is made up of 30S. Clear with the concept? In the prokaryotic cells, 70S type of ribosomes are going to be present, and in the eukaryotic cells we can find the 80S type of ribosomes. Yes. See here, this is also an important statement just now whatever we have discussed. These are the round bodied structures present freely in the cytoplasm or attached to the ear. If they are present freely in the cytoplasm, then we can observe this nature in prokaryotic cells, and when they are attached to the ER, that is nothing but endoplasmic reticulum, then we can observe these case in the eukaryotic cells. So, once please try to recall the previous session, so by that it becomes easy to all of you students. Yes, as I said you in the first introduction itself, this chapter and this first chapter, the cell, the fundamental unit of life, it is very important for your examination point of view. From this chapter only you are going to get approximately 30 to 40% of the weightage. From this chapter, each and every question you are going to get in the examination. So, please try to be patient and try to understand the concepts, my dear students. Yes. Next cell organelle is lysosomes. Lysosomes are called as, what we call for the Golgi bodies, they are called as postmans of the cell, and then next when it comes to the nucleus, it is called as control center of the cell, or else it is also called as the brain of the cell. So, now coming towards the lysosomes, these are called as the suicidal bags of the cells. These are called as suicidal bags of the cells. Yes. Why we are calling these particular ribosomes as the suicidal bags of the cells? Now, let us discuss about it in detail to make you the concept in a clear understanding way. Yes, the lysosomes are also the single membrane bound cell organelles. These are the single membrane bound cell organelles. We can observe some granular like structures inside these lysosomes. There are some granular there are some structures, granular like structures which are present inside these lysosomes. Yes. So, the scientists who discovered these lysosomes are yes, this is also very important question, children. You can expect it in the level 1 type of questions. The lysosomes was discovered by the scientist Christian de Duve in the year 1955. He is the scientist who discovered the lysosomes. These are also the single membrane bound cell organelles which are actually involved in digesting the cellular waste materials. So, see here, these are also the single membrane bound sacs present in the cytoplasm. These are going to be remained freely in the cytoplasm. Okay, and these lysosomes are going to contain few important enzymes. Many enzymes are there, up to 40 or more than 40k enzymes are going to be present. So, apart from that, few most important enzymes which are present inside these lysosomes are acid hydrolases. Acid hydrolases are the enzymes which are present in these lysosomes, and these enzymes are active in the acidic pH 4 to that is nothing but 4.5. So, inside the lysosomes we have the we have some digesting enzymes, those are called as the acid hydrolases. What does the term hydrolases means? Hydrolases. So, hydrolases are nothing but not just splitting the water. So, it is nothing but just adding the hydrogen ions inside to this lysosome cell. So, this addition of the H plus ions, the addition of hydrogen ions, okay, the pumping or else the movement of the just giving the hydrogen ions into these lysosomes, it is going to make the active. Okay, only in the presence of hydrogen ions only these particular enzymes are going to remain in active state, and the activity of the pH is 4.5. Okay, the main important enzymes present in the lysosomes are acid hydrolases. It is very important. Yes, and coming towards this most important functions, just the lysosomes are the cell organelles which are going to ingest, which are going to just break down all the cellular waste materials. They can also digest the bacteria, pathogens and some of the few worn out cell organelles. It can digest the broken down cell organelles, and it can also digest few pathogens like bacteria. It is just it is called as a suicidal bag of the cell just due to the function that it is going to break down and it is going to just ingest all the cellular waste materials. It is going to just break down the cellular waste debris. That is the reason these lysosomes are called as the suicidal bags of the cell. Yes, and next one more important thing about the lysosomes is it is going to exhibit the polymorphism. What does the term polymorphism means? Yes, we will look into this. Yes. So, polymorphism in the sense the lysosomes may exhibit in 4 forms: primary structure, primary lysosomes, secondary lysosomes, and the next residual bodies and autophasic vacuoles. So, when you just recall the process of exocytosis and endocytosis in the transport mechanisms of the plasma membrane, there already we have studied about the lysosomes in a short introduction format, children. Okay, there the particles, actually the plasma membrane is going to invaginate the food particles, later that food particles is going to be formed in the vacuoles, later that food particle along with the vacuole is going to fuse with the lysosomes. After fusing, the undigested residues are going to be just sent out from those cell membrane, that is the exocytosis and endocytosis. So, now based on that only we are going to learn about the primary structure of the lysosomes, secondary lysosome, residual bodies and the next autophasic vacuoles. So, please look into this, by that it becomes these terms clear for all of you. Yes. See, here is a food particle. First the lysosomes are, the first formed lysosomes are called as the primary lysosomes. The first formed, the freshly formed lysosomes are called as the primary lysosomes. Just imagine this is as a cell. Inside the cell what we have? Cytoplasm. This is the yellow colored structure, whatever I am drawing, this is just I am naming it as the cytoplasm, and inside the cytoplasm only we have the lysosomes. The lysosomes are only present inside the cytoplasm. And look into this. See, when the acid hydrolysis are going to release into this cytoplasm, what happens? Suppose just imagine here we have one more cell organelle, mitochondria. Okay, and then next here we have the chloroplast. It is just imagine it as a typical eukaryotic cell, and in the cytoplasm we are going to find the freely suspended single membrane bound cell organelles called as the lysosomes. And inside the lysosomes what we have, students, what are the most important enzymes? Acid hydrolases. The acid hydrolases are active in the pH of 4.5, and the activity of the activity of that acid hydrolases is just due to the presence of the hydrogen ions. It is nothing but these lysosomes can break the cell's waste. That lysosomes can break the cellular waste materials only in the presence of water. Only in the presence of water it is going to break the cellular waste material. That is what we are calling it as acid hydrolases. Clear? Yes, here we have the lysosome. So, when this lysosome is going to release these acid hydrolases, what happens inside the cytoplasm? If it is going to release the acid hydrolases, just it is going to damage all the cell organelles, right? So, this is the process, but later what happens? This is the plasma membrane. See, it is the plasma, just imagine this process is going to take place in amoeba, as we have studied in the endocytosis and exocytosis. See, this is going to be the plasma membrane of the amoeba. Here is a food particle. So, the primary lysosomes are the first formed lysosomes. Those are freshly formed or else newly formed lysosomes. That is called as the primary lysosomes. I am writing it here. Primary lysosomes. The primary lysosomes are newly formed or freshly formed lysosomes. Newly formed or freshly formed lysosomes are only called as the primary lysosomes. Yes. Now, that primary lysosome is going to fuse with vacuole. That primary lysosome is going to be fused with the lysosome. See, look into this. Here is a food particle. This plasma membrane is going to be invaginate to this. After invagination, here we are going to find the vacuole. Yes. Just I will draw it here, children. So, by that it becomes easy to you. Yes. The first newly formed lysosomes are called as the primary lysosomes. So, next later, if it is going to take the food particles, okay, actually we are calling it as the suicidal bags, just as it is going to break down the cellular waste materials. Yes, the secondary formed lysosomes, the secondary lysosomes are when these lysosomes will fuse with the vacuole along with the food particles. This is one vacuole where it is going to contain some food particles. Now, this vacuole along with the food particle is going to be fused with the lysosome. This is called as secondary lysosome. I am writing it as S and L. So, the primary lysosomes are first formed or newly formed lysosomes. Later, after the food particle which is going to be included inside that vacuole, that food particle along with the vacuole is going to be fused with the lysosomes. So, hence here we are calling it as the secondary lysosomes. Clear with the concepts? So, now residual bodies. So, what does the term residual bodies means? See here, the food vacuole is going to be fused with these lysosomes. So, later all the food which is going to be present inside that vacuole, that lysosome is going to break down that food molecules into the smaller substances. It is going to break down the food which is present inside that vacuole. So, after breakdown, the undigested food particles, whatever going to be present inside that, they are going to be sent out. So, that is called as residual bodies. So, the residual bodies are simply mean that the undigested food particles or else the undigested cellular waste materials. See, these are called as the residual bodies. Clear with the primary, secondary and residual bodies? In the primary lysosome, yes, primary, secondary and residual bodies, these are residual bodies, these are primary and secondary lysosomes. The primary lysosomes are first formed or newly formed lysosomes. So, next later these primary lysosomes are going to be fused with the food vacuoles. After fusing, that food which is present in the cell, it is going to be break down into the simple substances, and then next after that breakdown, the undigested food molecules or else the undigested food particles are going to be sent outside from this lysosome. This is actually called as the we can also call it as endocytosis. Okay, from here whatever it is going to be fused, it is called as endocytosis, and these residual bodies after the undigested food whatever it is going to be present, if it moves from this cellular external material, that is nothing but from the plasma membrane, this is called as the exocytosis. Endocytosis means cell eating process, and exocytosis in the sense cell vomiting process. This already we have learned. Yes, here are the residual bodies, undigested residual bodies. Now, just imagine this as a plasma membrane. Yes. So, this all are going to be come out from this. These are again just freely released to the external cell environment. So, now next coming towards the autophagic vacuoles. So, autophagic vacuoles, that is nothing but the vacuoles which are going to contain the cellular substances. Suppose just imagine it is as a mitochondria, yes, and this mitochondria is damaged due to some metabolic activities. This mitochondria is damaged and it is not going to show the perfect functions when it is previously. Okay, when compared to the previous functions, now these mitochondria, due to that breakdown, it is not going to show some proper and correct functions. So, at that time, here again it is going to be surrounded with the vacuole, and then next the lysosomes will fuse with this cellular components. The lysosomes next fuse with these vacuoles which contains the cellular components or cell organelles, and then next again these lysosomes will release the acid hydrolysis, and then next it is going to break these particular cell organelles. This is actually called as the autophasic vacuoles, which is going to contain the cellular substances, and the cellular substance along with the vacuoles will fuse with the lysosomes. There that lysosomes is going to break down, and the next that lysosome is going to be pushed out, and it will release the worn out cells, and it will create the new cells. See, for each and every cell it is going to have the lifespan. After some proper functioning, after some days, the older cells are going to be worn out, and that older cells are going to be replaced by the new cells just by the process of digesting its cellular waste debris due to the presence of the lysosomes. These are actually the functions performed by the lysosomes. Yes. Now we will look into the exact functions of the lysosomes. See, intracellular digestive system. We are calling the lysosome as the intracellular digestive system, and next we are also calling it as the suicidal bags, because it is going to digest the cellular waste materials, and then next it is also going to worn out the poorly working cellular organelles. As I said that if there is any disturbance in some cell organelles, if it is not going to show the proper functions when compared to the previous ones, then that particular cell organelle will form a vacuole around it. So, now that particular vacuole we are calling it as the autophasic vacuoles. Then later that autophasic vacuoles will fuse with the lysosomes, that lysosomes will break those cellular components, and then next new cells are going to be formed due to the presence of these lysosomes only. Yes, hence it is called as, due to these all functions only it is called as, sometimes it is also called as the garbage disposal system of the cell. These are actually the functions of the lysosomes. We are calling it as garbage disposal system, and we are calling it as suicidal bags, and next we are calling it as the digestive bags, because all these processes are going to be come under one statement, because it is going to worn out the poorly working cells, and it is going to digest the cellular waste materials. Yes. So, now next coming towards the one more important cell organelle, mitochondria. You all have some basic idea about all the cell organelles, students, because already all people have studied in your 8th standard, but here you are going to learn some interesting functions about it. That is the only difference. So, yes, see here, this is a mitochondria. So, mitochondria is also called as, what it is also called as? It is also called as the powerhouse of the cell. The mitochondria is also called as the powerhouse of the cell. Why we are calling that mitochondria as a powerhouse of the cell? Just look here. See, students. Yes. First we will learn about the discoveries. Who discovered this particular mitochondria cell organelle? Yes. Just a minute. Yes, clear. So, the mitochondria are first seen by the scientist Colliker. Mitochondria are first seen by the scientist Colliker. This is also very most important question which is going to be appeared in the some case study questions as well as in the section A type of questions. Yes, mitochondria are first seen by Colliker in the year 1880, where he observed those mitochondria in the muscle cells. In the muscle cells, Colliker has seen the mitochondria. Later, Benda, the scientist Benda, named them as mitochondria. So, he seen the mitochondria in the muscle cells in the year 1880, and then next the scientist Benda has named those structures as, those cell organelles as the mitochondria. Yes. Now, we look into this particular structure of the mitochondria. Yes. See, look into the diagram properly, students. Yes, from the nucleus, when we started the nucleus subtopic, from the nucleus all the cell organelles are very important for the 4 marks as well as 5 marks. They may ask this question for to mention the diagram as well as its function. So, it is mandatory to learn all the diagrams and it is mandatory to remember all those labeling parts along with the functions also. So, this mitochondria is also one of the PYQ only. In the previous year questions, this question is also mostly asked, which organelle is called as powerhouse of the cells, and mention it with a neat labeled diagram along with the functions. This is the actual question which you got in the PYQ. Yes. So, now we will look into this structure. The mitochondria is also a double membrane bound cell organelle. The mitochondria is also a double membrane bound cell organelle. It is going to be present, yes, it is going to be present in the eukaryotic cells. Yes, it is a double membrane bound cell organelle. This is, yes. So, this is the outer membrane, yes, this is the outer membrane, and next here we have the inner membrane. The mitochondria is double membrane bound cell organelle which contains the outer membrane as well as the inner membrane. In between the outer and inner membrane there is a space filled, that is called as the perimitochondrial space. Perimitochondrial space. So, the perimitochondrial space is the space which is going to be present in between the outer and inner mitochondria. If we observe into the inner side, the inner membrane is going to be shown some finger like projections. It is going to be folded many times and it is going to give the appearance of some finger like or else some folded like appearance. So, this is called as cristae. See, this will be the outer membrane, yes. And the next inner membrane, can you see this? This is the inner membrane which is going to show some many overtones. This is called as cristae. Okay, the cristae, the presence of these folding structures, this cristae is involved in increasing the surface area of the cell organelle. The multiple foldings of the cristae will increase the cell organelle. It will increase the surface area of the mitochondria, and the fluid substance, the substance whatever it is going to be present inside these cristae, it is called as the matrix. Can you see all this yellow colored substance? That is called as the matrix which is present inside this inner membrane, and this is called as the cristae. So, like DNA, this mitochondria is also going to have its own genetic material in the circular form. Mitochondria also have the own genetic material in the circular form. It is called as the DNA. Yes, this is also one of the most advanced character. Okay, yes, and chloroplast is also going to have its own genetic material, that is DNA, and this mitochondria is also going to have the ribosomes. Mitochondria are also going to contain the ribosomes, hence they are also involved in some protein synthesis. If you look into this inner membrane, yes, if you look into this inner membrane here, yes, here you can find some particles called as the elementary particles. The elementary particles are present inside the inner membrane. Actually, they are going to be in this form. Yes, I will write it here, children. This is also very important. So, please just note down. The elementary particles are going to be present inside the inner membrane of the mitochondria, and that elementary particles are going to be attached to that inner membrane. It will be in this form. Yes, these are called as elementary particle. I am just drawing the single structure, how actually the single elementary particle looks like. See, this will be the base, and yes, see, can you see here? This is called as F0 portion, and then next this is called as F1 portion. Yes. F1 and the next F0. F1 and F0 are the elementary particles which are present in the inner membrane of the mitochondria. So, this outer membrane is selectively permeable membrane and the inner membrane is semi-permeable membrane. So, in between them we have perimitochondrial space. So, the fluid or else the solvent whatever present inside in between the outer and inner, easily the solutes or solvents can enter from this inner membrane to this, yes, from this outer membrane it can also enter to this inner membrane, and from here it is going to reach to the cristae, that is nothing but it is going to reach to the matrix. So, now these F0 and F1 particles are called as the elementary particles. So, inside these cristae, inside this matrix, there are several enzymatic reactions which are going to takes place. Several reactions will occur inside the matrix like Krebs cycle. So, the Krebs cycle is involved in the cellular respiration. The Krebs cycle is going to take place in the inner membrane of the mitochondria. It is involved in the cellular respiration. About cellular respiration also you are going to learn in the higher grades, you are going to learn elaboratively in the 10th standard, students. Yes, this Krebs cycle is going to be, it is actually an enzymatic reaction which will takes place inside the inner membrane of the mitochondria. Yes, now these are the F0 and F1 particles which are also called as the elementary particles. The mitochondria is actually involved in generation of energy. It is involved in generating the energy in the form of ATP. What is the full form of ATP? Adenosine triphosphate. Adenosine triphosphate. Yes. So, now this F1 particle, the F1 particle is inactive in state. So, we have to generate the energy in the form of ATP, right? That is actually the goal of the mitochondria. That is the only main function of the mitochondria. But here if you look into the elementary particles, the F1 particle is inactive. So, the F1 particle is going to become active when we are going to supply the hydrogen ions. When we pump the H plus ions, when we push and when we pump the hydrogen ions, then only the hydrogen ions, hydrogen molecules or else the hydrogen ions will travel from here and it will reach to the F1 particle, and here they are going to combine with ADP, ADP plus PI, adenosine diphosphate, plus when it combines with one inorganic phosphate, then it is going to form the ATP. So, here in this process there is one enzyme involved, okay, the ATPase enzyme. ATPase. The enzyme ATPase is going to convert the adenosine diphosphate plus inorganic phosphate, and it is going to convert this ADP plus IP, inorganic phosphate, into ATP. So, by this is how actually the ATP is going to be generated. Okay, as we are saying it as outer membrane is selectively permeable and inner membrane is semi-permeable. So, semi-permeable means in the sense some solvent molecules may enter into the inner mitochondrial matrix. So, when some solvent molecules are going to enter at that particular point, these F0 and F1 are elementary particles which are going to be attached here. F0 and F1 are the elementary particles. So, in that F0 and F1 is going to remain in inactive state. So, it is going to become active when we pump the hydrogen H plus ions from the inner outer in between the perimitochondrial space we have no, from there we are going to transport the hydrogen ions, then this F1 particle is going to become active and it is going to convert the adenosine diphosphate plus inorganic phosphate into ATP. In between this there is one enzyme which is going to be involved in conversion of ADP plus inorganic phosphate into adenosine triphosphate. That enzyme we are calling it as ATPase. So, whatever the food we are eating, that food is going to be digested. Okay, the digested food is going to give the energy, it is going to combine with the cells and it is involved in giving the energy and it is involved in generating the energy. Okay, with the in the presence of oxygen, the glucose is going to be broken down and then next it is going to release the carbon dioxide plus water and energy. Yes. So, this food whatever we are going to consume, this digested food is going to combine with oxygen and then next it will release the carbon dioxide plus water plus ATP. This is the process how actually it is going to occur. Clear with the concept mitochondria? The mitochondria is also a double membrane bound cell organelle. It is going to contain the outer membrane and inner membrane. In between them we have the perimitochondrial space. The outer membrane is selectively semi-permeable and inner membrane is also selectively and semi-permeable to some solvent molecules. And inside the mitochondria, the inner membrane is going to show some finger-like projections and some many foldings. That folding structures are called as the cristae. Inside the cristae, there is a fluid-filled substance called as the matrix, and the mitochondria is also going to contain its own genetic material in the form of circular DNA. And then next, inside to this inner membrane, we are going to contain the elementary particles, those are called which is going to contain two components, F1 and F0. Okay, and that F1 is going to remain in active. Yes, the F1 is going to remain in inactive. When we pump the hydrogen ions, it becomes active by the presence of enzyme ATPase. So, in each and every cell, the number of mitochondria is going to vary. The mitochondria percentage and the mitochondria number is going to vary from each cell. If the cell is going to contain more mitochondria, then it contains more elementary particles, then it is going to release more amount of energy. If the cell is going to contain less number of mitochondria, then the elementary particles may remain less, and it is going to give less energy in the form of ATP. More the mitochondria, more will be the amount of ATP generation. This is how actually the process of generation of ATP is going to takes place. So, here you no need to study about the Krebs cycle, you are going to learn it in the higher standards children. So, inside these matrix only enzymatic reactions are going to take place. For that we have in your NCERT there is only one example, that is called as Krebs cycle. Apart from Krebs cycle, we also have the electron transport chain also. So, the Krebs cycle is actually involved in the cellular respiration process. So, now just we look into the functions of the mitochondria. Yes. See, mitochondria are first seen by Colliers, and these are present in the all the eukaryotic cells, but those are absent in the prokaryotic cells. Prokaryotic cells are always going to lack the double membrane bond and membrane bond organelles, and the outer membrane is permeable. Yes, inner membrane is selectively permeable. See here, cristae bears the ATP generating oxysomes or elementary particles. As we have discussed clearly, the cristae, inner membrane, inside the inner membrane we have the matrix. So, that cristae is going to contain the elementary particles. Okay, these are also called as oxysomes. So, inside them adenosine triphosphate generates during the respiration. During the respiration process, energy is released and it is generated as ATP, and it is required. Why it is required? For all the chemical activities which are needed for the life. Whatever, if you want to perform the activity, if you want to walk, if you want to learn, and if you want to do some exercise for doing each and every activity, we need the energy, and all that energy is going to be supplied from the cell organelle called as the mitochondria. This is actually how it is going to be wondered. Okay. So, this is the mitochondria, which is called as the powerhouse of the cell. And the next, see here, hence this is the reason this is called as the powerhouse of the cell, or else sometimes it is also called as storage batteries of the cells. Okay. Why we are calling it as storage batteries or powerhouse of the cell means we can do each and every activity. If we want to perform any activity, we require some energy, and that energy is going to be supplied from the mitochondria. Yes. Yes, once we will look into these functions, these are called as biochemical factories, and where food is oxidized and energy is released. Yes, the glucose which will combine with the in the presence of oxygen, it will break down that food and it will release the carbon dioxide plus water and energy in the form of ATP. Yes, it is also involved in the Krebs cycle and electron transport chain. This also we have discussed clearly just now, and it is also involved in the programmed cell death. Did you remember programmed cell death? It is also called as apoptosis. While studying the functions of lysosome, we have already discussed autophasic vacuoles are nothing but the vacuoles with some cellular components and cell organelles. Okay. If this mitochondria is going to be damaged, then it is going to be surrounded with the autophasic vacuole, then it is going to fuse with the lysosomes. Okay. Again these lysosomes will break that worn out cell organelles and it will replace this damaged mitochondria with new mitochondria. Yes. Yes. Clear students? Yes. Here is one interesting and important news to all of you, my dear students. Yes. Neet World, your step to medicine. This is our institute, Neet World, which is located in Narayanaguda, Hyderabad. Yes. So here is one interesting thing to all of you. So I request each and every student to utilize this particular opportunity. So we are starting the foundation courses. We have already started the foundation courses from the 1st April, but the second batch, okay, second batch for this foundation course is going to start from 15th April, and all the students of the classes 6th, 7th, 8th, 9th and 10th are going to be eligible for this particular foundation course, and the batch is going to start from 15th April. For every 15 days we are going to start the new batch, my dear students. Yes, so I request each and every student to join the Neet World to become higher, okay, to become the doctors. This is actually Neet World, your step to medicine. So in our institute Neet World, so the main specialty of our institute is we are going to cover the topics, we are going to cover each and every chapter which are mostly important for NEET and JEE mains. This is only our main motto. We are going to cover each and every concepts in a clear conceptual understanding way, and we make you to learn in better way by creating the animated videos. Okay, so by this particular thing you are going to get the clear crystal idea about this particular concept. Yes, and every 15 days we will start the new foundation batches, and this batch, this foundation summer camp is only for the classes 6th to 10th. It is a 15 days camp, my dear students. So in that 15 days, the first 10 days we are going to make you to learn the basics. We are going to show the concepts of the basics. So after 10 days, next 2 days. So the next 2 days we are going to make you to learn in the active type of the mode. Yes, these are activity based learning mode. So after completing those 10 days foundation courses with regarding to the basic concepts, next 2 days we are going to perform some interesting activities with that particular topics, and then next after those 2 activities, next we are going to conduct the quiz. In that 10 to 13 days, whatever we have teach you and whatever you learned, based on that we are going to conduct the quiz. That is just to test your talent, my dear students. And in the last 2 days we are going to conduct one assessment test. Okay, and then next, that is full length test, which is going to cover all the basics. All the basics whatever you are going to study in the first 13 days, you need to write the best lengthy wise attempt assessment test on the 14th day. In the last day there will be one interesting career guidance and motivation session with some expert faculties, my dear students. So this is all about our institute Neet World. So don't miss this summer camp foundation course opportunity, and I request each and every student to join in our institute, and we make you all the concepts in a clear manner by creating each and every concept in some animated videos, and the main motto of us is we are going to cover all the subjects like physics, chemistry, mathematics and biology in both media, like that is for SSC as well as CBSE. For both we are going to make the concepts clear. So yes, this is all about the Neet World Institute, which is located in Narayanaguda, Hyderabad. Hope you all are clear with this today's particular session. So we have completed the endomembrane system. Okay, we have completed the endomembrane system, and in the next upcoming session we are going to learn about the vacuoles, plastids, and then next some cell bodies and cell division, cell inclusions. These are the leftover topics from the chapter Cell, the Fundamental Unit of Life. So I request all of you to join to the next upcoming live session. Thank you. Please like, share and subscribe to Neet World.